So, chapter 32 then. It's a prelude to a survey of the animal phyla that we're going to look at. So I'm going to tell you a lot of terms, define them, describe them, and illustrate them that we're going to use when looking at the other phyla. Okay? All right. So, think about your kingdom. Yep, the animal kingdom. You're familiar with lots of animals, but you're probably not familiar with the vast majority of animal species that are out there. We tend to think of things that resemble us. Maybe they're cute and cuddly, or maybe, I don't know, snakes you might think of as animals. But worms are animals. Jellies are animals. Creatures that you would look at and just say, that looks like from outer space, can be an animal. All right? So we're going to try and survey all of that diversity in the animal kingdom so you're familiar with it. So we've identified about 1.3 million living species of animals. But what is our species count for the number of species that we've identified in total? How many species of organisms have we identified on the planet?
1.3 million?
Would you say?
1.8.
About 1.8 million species we've identified. There are about 1.3 million living species of animals, of organisms, of animals. Okay? So of that 1.8 million, that encompasses things that aren't animals and also extinct species. Okay? All right. So the diversity of animal life is breathtaking and overwhelming. It really is. And one place to look at that diversity would be a reef system. In fact, a fantastic place to teach this course would be Rocky Point. Got to tell you, not just because it's great in the evening, but in the daytime you get to go down to the tide pools, and that intertidal zone you can find many of the organisms that we're going to talk about. And there they are, living as they live, and they're live, and it's fantastic. But so, any scuba divers in the class? Awesome. So, reef systems have incredible animal diversity. All right, and you can go to these reef systems and you might see many, many different phyla in just a very small space, a few meters of the reef. So let's define an animal then. All right, let's see what makes an animal an animal. Now I'm going to give you a list of criteria that define animals, but no single definition defines animals. And what you'll find to most of the one-line definitions, there are exceptions. All right? So, when we look at many characteristics all taken together, even though there might be exceptions to each of those characteristics, when all clustered together, that defines what an animal is. So first, animals then are heterotrophs that ingest their food. So what does a heterotroph do, or what does it mean? It's got to eat something else. It can't make its own energy-rich molecules. It's got to consume energy-rich molecules, usually of another organism, to get its energy and the other things that it needs to live. But a fungus is also a heterotroph. Right? It also feeds on other organisms, can't make its own energy-rich molecules. So what distinguishes you from a fungus in terms of feeding? The way they feed, yeah. We ingest our food. Fungi are heterotrophs and take in the nutrients by absorption. Typically, a fungus would release enzymes outside the cell. The enzymes would break down whatever substrate it's on, and then it would absorb those breakdown products. But you ingest them. That means most animals, not all of them, but it's a pretty good rule, have a mouth. Ingest through a mouth. Okay. All right, so what would be a contrast to heterotrophic nutrition? What would be a contrast to a heterotroph? An autotroph, right? Plants are autotrophic. They're able to make their own energy-rich molecules, of course, using sunlight energy. It's just a photosynthesis and energy conversion process. Yeah?
So So do giant tube worms count as animals?
Yeah. Yeah.
But they don't digest food.
So, again, there's lots of exceptions. Some organisms don't have a mouth at all. Tapeworms don't have a mouth at all, so they do absorb. So there are exceptions to these rules. Yeah. All right. So animals then are multicellular eukaryotes. The individual is made of more than one cell. They're multicelled eukaryotes. You know what a eukaryotic cell type is, don't you? Yeah. Okay. And they don't have cell walls. All right? Animal cells don't have cell walls. In fact, most other organisms, most of the other kingdoms and domains, they do have cell walls. All right? The plants do, the protists do, the fungi do, and so do a lot of the protists. All right. What do animals have that's kind of not quite a cell wall, but similar-ish? What do we call it? Every cell has a cell membrane. Every single cell has a cell membrane. A cell wall is not instead of a cell membrane. Plants have a membrane and a cell wall. Fungi have a membrane and a cell wall. Animal cells have a membrane, no cell wall. What do they have exterior to the membrane, which is, it's not really a cell wall, but you can think about it as almost analogous to extracellular matrix, which has collagen as part of it. Good. All right. So, animal bodies are held together by structural proteins. Many of these other organisms, their bodies and their biomass is kind of held together by the cell walls being attached to each other. Your bodies are held together by structural proteins. One cell is connected to another cell by these structural proteins. Right? Muscles and ligaments have their strength from these structural proteins. An example of structural protein is collagen. What would be an example of another structural protein? Keratin. Good, keratin, yeah. So animals then have nervous tissue and muscle tissue, and they seem to be absolutely unique to animals. And I'd say the only exception is maybe the sponges don't seem to have sort of true nervous or muscle tissue. But then, I don't know, taxonomy goes through lots of revisions, and right now we're even wondering whether sponges should be animals or whether they're in a different group. Not to date myself, but when I was in high school and we did taxonomy, it was easy. It went like this. There are two kingdoms, plants and animals, and every organism is one or the other. And then it changed. Then it said, all right, we need to change that. We need to have 5 kingdoms. Plants, animals, fungi, protists, and bacteria. Now we've changed it again. We've got 3 domains. Okay? So it goes through changes. Animals have tissues, true tissues. All right? And again, slight exception to that are the sponges that don't really have true tissues, but animals have tissues. And they're groups of cells that have got a common structure and function, or both. And tissues, cells of tissues tend to occur in groups, and they're often separated by membranes. Often they occur in layers. So most animals reproduce sexually, although you've got many animals that undergo asexual reproduction. And the diploid stage usually dominates the life cycle. So what do we mean by the diploid stage? What does that mean? What does diploid refer to? It's related to mitosis, it's part of it. But what does it refer to? You're on the right tracks. What would be the opposite to diploid? Not the opposite, but perhaps the haploid. Right. So remember, your chromosomes come in pairs, homologous pairs. If you're diploid, that means you have one each of all of the pairs. If you're haploid, you have one of each pair. Gametes are haploid, typically. Right? You, as an individual, as an adult, are diploid, and most animals are diploid, or at least that phase dominates the life cycle, the diploid phase. So after a sperm fertilizes an egg, what results is a zygote, and that undergoes cleavage. think about that as a rapid cell division. The zygote's a single cell, that rapidly divides. And there are certain patterns of cleavage that animal groups go through. It's very predictable. And I'll show you pictures of those, what they look like, and you can see the different cleavage types under the microscope. So cleavage is a cell division. If you get one cell and it divides once, you've got two cells. If each of those divide once, you've got 4 cells. And then let's keep going, lots of cell division, and you end up with a ball of cells. And that ball of cells eventually hollows out, so it's hollow in the center. It is essentially, you can almost think about it as like an orange, but just the peel, and that's a single cell layer, hollow in the middle. And we call that a blastula. Now I'll show you some images of that in a moment. And then the blastula further develops in a predictable way. It undergoes something called gastrulation. And rather than describe it now, I'll describe it with pictures. After gastrulation, it forms something called a gastrula. And it's this development, this folding at gastrulation that forms a gastrula. Looks nothing like you. It's essentially just a big mass of cells. But then we start to set up different embryonic layers. All right? And that's characteristic of animals. And all of your tissues and organs are derived from those different embryonic tissues or embryonic layers. So I'm going to refer to these embryonic layers frequently. I'm going to refer to them a lot. Okay, so here we've got a zygote. Single cell results from a sperm fertilizing an egg, and then we're going to undergo some cleavage. So how many rounds of cleavage would it take to get this, the 8-cell stage? How many?
4.
It's not a trick question.
3.
3. Okay. And you can actually see them. There's one plane of division. There's another one. One division makes 2. Second division makes 4. Third division makes 8. So this is the third division here. Now when we have the first and second division, typically we get 4 cells that are very similar in size. But at that third round of cleavage, now on the top here, we've got smaller cells than on the bottom. That's important. Yeah.
Is cleavage, rapid cell division exclusive to zygotes, or just anything that would go through rapid?
No, you can think about cleavage as being the cell division that a zygote goes through during that very early round of cell divisions. Cleavage.
So it's only used to define that?
Only in this context. We wouldn't talk about cleavage in you or your liver, for example, right now.
Okay.
Yep. All right. So let's undergo more cleavage, more cell division, and we end up with a blastula. Looks like this, which is a hollow ball of cells. And the cavity inside is called a blastocele. So this term cele is often used to refer to a cavity. All right. And you can already see in this blastocele that not all the cells are exactly the same size. All right. So now we're going to undergo this process of gastrulation. So you can always think of gastrulation if I take a hollow ball of cells and now I'm going to push in a region. It's like taking a balloon and pushing in with your thumbs. Yeah. So you're going to push some of those cells in. And there's nothing pushing them in, by the way. They're just kind of migrating. All right. There's processes that pull them in, but there's not a structure that pushes them in. Okay. So the process of gastrulation is where you get that invagination. Cells get pushed in. And at the end of gastrulation, we've got something like this. It's a ball of cells, and we've got this outer layer of cells, and then we've got this inner layer of cells. and we've got a cavity here which exits to the outside, and we've got a cavity here which has no exit to the exterior. Is everybody okay with that? Now, the minute we get these layers, we can give them a name. This outer layer of cells is called ectoderm. Ecto means outside, derm refers to skin, outside skin. It's not really a skin. So this layer of cells is called the endoderm, inside skin. The cavity between the ectoderm and endoderm is called the blastocele. And then this cavity here, the one that opens to the outside, is called an archenteron. That's quite a lot of new terminology, isn't it? But the ectoderm should be pretty straightforward. Endoderm, pretty straightforward. So archenteron. And the little pore that connects the archenteron with the outside is called the blastopore. Okay? Everybody okay with that? So we call these germ layers. Ectoderm and endoderm are germ layers. And it's from those layers that many of your tissues and organs differentiate down the line. For example, your skin tissue differentiates from ectoderm. Your digestive system differentiates from endoderm cells. Okay? Now, if you were a layer of cells between the endoderm and the ectoderm, what do you think you'd be called? What was it? Mesoderm. Meso means middle skin, mesoderm. This diagram doesn't show mesoderm, but mesoderm develops in some phyla, the majority of them, in fact. All right, so let's see if we can watch a video of gastrulation. How do I just say no, go away?
The egg cell starts to divide.
Thank you.
8 cells, and so on. Continue. Soon after fertilization, the egg cell starts to divide into 2, 4, 8 cells, and so on, continuing to double the number of cells with each division. During this time, there is no increase in the mass of the cells. A circular ball of cells is eventually formed, called the morula. It gives rise to the blastula, and after further development, to the gastrula. As a result of the movements that take place during the formation of the gastrula, 3 embryonic germ tissue layers form: the ectoderm, mesoderm, and endoderm. From these tissues, all the animal's organ systems develop. In addition to these germ layers, humans and other mammals develop 4 extra embryonic membranes. These are called the amnion, the yolk sac, the allantois, and the chorion. They provide the developing embryo with protection, nutrients, waste exchange, and regulation of gases and air.
So we'll talk about those extra embryonic membranes when we talk about chordates. Right. Did that help? Did that animation help to see how gastrulation occurred, that process? Yeah? All right. So all animals go through that process. So many animals have got at least one larval stage. Okay? We don't. But many animals do have a larval stage. And I want you to think about a larval stage as a sexually immature stage. And it's usually, morphologically, quite different to the adult. Now again, there's lots of exceptions to that. But think about how a caterpillar is different to a butterfly. Caterpillar's the larval stage. And in order to turn into adults, sometimes they go through metamorphosis. Think about a tadpole as a frog. That's considered the larval stage. Goes through metamorphosis, and you get a frog. Lots of exceptions to that, but it's a generality. So the juvenile stage of an animal can resemble an adult, or it can look very different. But what differentiates the juvenile stage from the adult stage is that the juvenile stage is sexually immature. The adult stage is sexually mature. And again, there's a couple exceptions to that, with some salamanders that at juveniles can sometimes become sexually mature. Okay, so let's have a look at these phylogenetics of animals then. Here we've got other eukaryotes. There's a branch that goes off here to the choanoflagellates and other protist-like organisms. And here we've got a branch, a very distinct branch, which leads to all of the animals. Right? And the very first divergence is to the sponges, and the other branch goes to all of our other animals. All right? So, when we're talking about animals, we're talking about the sponges and all of the other phyla that we're going to look at. Some of these exceptions apply to sponges. They diverged so long ago that there's not a lot of distinction between protists, or at least a colony of protists. So that distinction's a little bit vague. Okay, now we're going to talk about body plans. All right? So I want you to think about the body plan of an organism, and we've got lots of different terms to describe different aspects of the body plan. Okay? So, one aspect of body plan is the symmetry that an organism has. So symmetry is one component of the body plan of an organism. So most animals have some kind of symmetry. Some don't, they're asymmetrical. But usually an animal group will have a specific kind of symmetry. So for example, some animals can have radial symmetry. And they don't really have a back and a front or a left and a right, but they're symmetrical about a diameter or sometimes even a radius. Okay? So some organisms have radial symmetry, and I'll show you a diagram of that. What's this organism? It's an anemone. Good. So an anemone has radial symmetry. It doesn't really have a left and a right or a back or a front. It does have a top and a bottom, and you can make that as an analogous to a flower pot, okay, which doesn't really have left and a right or a back and a front, and that's got radial symmetry. Now, in contrast to radial symmetry, we've got bilateral symmetry. If you've got bilateral symmetry, you do have a distinct left and a right, and a back and a front, and almost always a top and a bottom. Okay? And if you don't have any symmetry at all, there is no plane of symmetry, you're neither radially symmetrical or bilaterally symmetrical, how do we describe your symmetry? Asymmetrical. Good, you've got asymmetry. What about this guy? What is it, first of all? Sand dollar. How would you describe its symmetry? Got three choices. Bilateral, radial, or asymmetrical? Bit of a tough call to make, isn't it? So I don't want you to think of every organism as being like perfectly symmetrical all the time. All right? But this one does have radial symmetry. But do you notice it's got 5 parts? So we can be a little more descriptive with this. We can say it's got pentamerous radial symmetry. Okay? What kind of symmetry does sunflower have? Radial. Yeah, pretty good radial symmetry. Again, not every segment is a perfect mirror of every other segment. What about this guy? Radial symmetry, for sure. There's another whopping great big sea jelly. The size of that, would be awesome to encounter that. Yeah, that's also got radial symmetry. Okay, two-sided symmetry, bilateral symmetry. You've got a left and a right, and a back and a front, and almost always a top and a bottom, a dorsal or a ventral surface. So bilaterally symmetrical animals have a top, which we refer to as the dorsal side, and a bottom, which we refer to as the ventral side. And they've usually got a distinct left and a right because they've got a distinct top and a bottom and back and a front. So we often refer to the front of the organism as its anterior end, and that's usually the head end, and the back as the posterior end, and that's usually the tail end. So you okay with these terms so far then? Radial, bilateral, dorsal, ventral, and then anterior and posterior should really be familiar with. So some animals have cephalization. And cephalization, think about that, it's the development of a head, or tending towards having a head-like end. Okay? That's cephalization. And there's different degrees of cephalization in different organisms. You've got an extreme degree of cephalization. If we take a tapeworm, not so much. It does seem to have a head end. An annelid worm, an earthworm, definitely has some cephalization. It has a head end. Head. Kinda. All right. Radial symmetry, bilateral symmetry. You good with those? So I'm going to use these terms to help describe the body plans of organisms. And certain phyla can be diagnosed in part by their symmetry. So organisms with radial symmetry then, often sessile or planktonic. What does sessile mean? Doesn't move very much, oftentimes not capable of locomotion. So they might be attached to a substrate, or if you're planktonic, maybe you drift, but not capable of too much locomotion. Some of them can be. And animals with bilateral symmetry are often a little more active, at least in the adult stage. They've got a central nervous system, which is used to sense the surroundings. They can coordinate movement. Usually they've got mechanisms of either propulsion or locomotion, at least as adults. Sometimes the larval stages don't. Okay, so animals then have tissues. And think about tissues as these collections of cells. Usually the cells are fairly specialized to perform a certain function, oftentimes distinct from other tissues. And they're usually separated from other tissues by membranous layers. All right? So that's true throughout almost all the animal kingdom. Again, there's a couple of exceptions with sponges. Sponges don't seem to have true tissues because you've got several different cell types in the same layer, and they're not separated by membranous layers. So again, the distinction's a bit blurred. So we've talked then about embryonic development, early embryonic development, giving rise to, what are the three embryonic germ layers? Ectoderm, mesoderm, and endoderm. Good. And it's from those embryonic germ layers that all of your organ systems develop. Okay? And we can look at almost every organ system in your body and trace it back to one of those three embryonic germ layers. Now, do you as an adult have ectoderm? Be brave. You don't. Because it's a layer that covers an embryo's surface. You as an adult don't have mesoderm or endoderm. Okay? They're only present in the embryo and usually early embryonic development. And it's from those layers that your organ systems develop. So you've got tissues and organs that are derived from ectoderm, derived from mesoderm, and derived from endoderm. Okay. So sponges then, and a couple of other groups, don't have true tissues. We still call them animals. They loosely fit the definition of animals, but they don't have true tissues. Okay, so some more terminology then, and this has to do with the number of embryonic germ layers that an animal group would have. So if, as an embryo, you have 2 embryonic germ layers, that's ectoderm and endoderm, then we refer to you as diploblastic. Okay? And that mostly includes the cnidarians and comb jellies. They're the groups that are diploblastic. It's pretty much the only group that is diploblastic, has 2 germ layers.
So starfish?
Starfish are echinoderms, all right? And they're actually quite advanced in terms of how far up the evolutionary tree they are. Okay. All right, so if you're diploblastic, you've got 2 embryonic germ layers, endoderm and ectoderm. If you've got 3 embryonic germ layers, ectoderm, endoderm, and mesoderm, how would we describe your layers? Not diploblastic, but triploblastic. Yep, good. So triploblastic animals then have got a mesoderm layer, cells between the ectoderm and endoderm, and develop into their own set of organs and organ systems. Most animal phyla are triploblastic. All right, so symmetry was one aspect of an organism's body plan. The germ layers you've got, I want you to think about that as being another aspect of an organism's body plan. Okay? Symmetry, what kind of symmetry can you have? Radial, bilateral, or asymmetry. Germ layers, what could you possibly be? Diploblastic or triploblastic. Now, if you're a sponge, you don't have true tissues, do you? So diploblastic and triploblastic don't really apply to a sponge, to the peripherans. Okay? All right. So now we're going to look at the third component of body plan, and that's the body cavities. So most triploblastic organisms have got a body cavity. And a true body cavity is called a coelom. And the coelom, or coelomic cavity, is derived from mesoderm. Now again, I'll show you some diagrams that will help you distinguish among these different body cavities. So if your cavity, which is a coelom, develops entirely from mesoderm, then it's a true coelom, and you are considered a coelomate. So what about if you're an organism that doesn't have a body cavity at all? If you've got a true coelom, you're a coelomate. If you don't have a true body cavity, no cavity at all, what do you think the term is there? Acoelomate. If you don't have symmetry, you're asymmetrical. If you don't have a true coelom, you're an acoelomate. Okay? All right, so let's have a look at this diagram then. We're going to use, in this case, a planarian. Little flatworm, we're going to look at them next week in lab. It's in the phylum Platyhelminthes. They're really, really cute. and we're going to chop them into little bits and watch them regenerate. But a flatworm is triploblastic, 3 germ layers, ectoderm, mesoderm, and endoderm as an embryo. So therefore, it's got organs and tissues derived from those 3 layers. But there is no cavity inside the organism. And so if you don't have a cavity, you're called an acoelomate. Okay? Now, I'm going to jump up to coelomate. We're going to use an annelid worm, an earthworm, as a good example of a coelomate. And the coelomate is a cavity that develops from within the mesoderm layer and organs and tissues that are derived from mesoderm. Okay? So you have a look at this diagram. There's your coelomic cavity, and you are entirely within tissues and organs derived from mesoderm. Does that make sense? So if I'm in a true coelom, everything I touch is going to be a tissue or an organ derived from mesoderm. Okay? Now let's contrast that to our third term, and that is a pseudo-coelomate. Pseudo means what? False. It's got a false coelom. There is a cavity, but the cavity is between tissues and organs that were derived from mesoderm and tissues and organs derived from endoderm. So if I am in a pseudocoelom, on the inside I'm going to touch organs and tissues derived from endoderm, and on the outside organs and tissues derived from mesoderm. Is everybody okay with those 3 terms? Yep. So the Platyhelminthes, the flatworms, are acoelomates. The nematodes are pseudocoelomates. Almost everything else are true coelomates. You are a coelomate. You have a body cavity, all right, which develops from within the mesoderm. Good? So, you can use these 3 body plan criteria to help define different phyla. So let's take maybe the phylum, the Platyhelminthes, the flatworms, okay, which we're going to look at next week. Did that flatworm have any symmetry? Do you want me to go back to its picture? Yeah. There, have a look down the bottom. There's the flatworm. What would you say its symmetry is? Good, bilateral symmetry. What about its germ layers? Triploblastic. And what would you say its body cavity is? Acoelomate. So, if you are bilaterally symmetrical, are triploblastic and an acoelomate, that pretty much makes you one thing and one thing only. A Platyhelminthes. Something in the phylum Platyhelminthes. So you see how we can use these to define phyla. And I'm going to want you to know the defining characteristic of all the phyla that we survey. Okay. So that's chapter, what was it, 32? That's wrapped up chapter 32. What I did is I went over all the descriptive words that we're going to use, well not all of them, but a lot of them. And I just thought I'd end with this picture. This is from a study abroad to Africa. Look, we got to see and touch cheetahs. Isn't that crazy awesome? They don't let you do that anymore. We cannot do that anymore. There was apparently some cheetahs attacked somebody in South Africa, and so that's it. They just made a rule now. You can't. You've got to have a fence between you, which is terrible, really, but